My name is Victor Roman. I am an undergraduate student at the University of Puerto Rico in Bayamon. I have been working on Dr. Behra's lab for this summer. My major is biology with a human approach, and I have loved science since very early. At first I wanted to be a veterinarian, and then I had this crisis in high school where I did not know what I wanted to be. Then I found a passion for healthcare, and I decided to be a doctor, to pursue an NMD. Ever since I entered the Choose Development program, I have really liked doing research, and the fact that all the hard work that we're doing here in the lab may contribute to the scientific community and improve medical treatment. The reason why I chose development is that I took a developmental biology course and I really liked the laboratory and all the different types of animal models that we were studying in that class. For some reason, we did not speak too much about zebrafish. When I saw that there was an opportunity to work in a developmental biology lab, I decided to go for it. Once I saw they had the option of working with zebrafish, I knew I had to choose that one.
Hi, my name is Valeria de la Rosa. I'm a graduate student here in the Medical Science campus at University of Puerto Rico. We are in the Dr. Martine Behra's lab. We use the zebrafish as an animal model. It's an excellent model because it has a genome's full sequence, has external fertilization, the embryos are clear so you can look the fish development. You can easily manipulate it with so many tools that has been developed. The focusing here in this lab is about hair cell regeneration. We want to elucidate how this zebrafish regenerates the hair cells. We have several projects around the regeneration. One of them is the one that Victor is explaining in this video.
My project for the summer is genotyping and establishing 8 stable mutant lines using the zebrafish as a model, which my mentors Dr. Martine Behra and Luis Colon, a graduate student, have generated using the CRISPR-Cas genome editing tool. We targeted known and novel genes, which we think are important during the development and regeneration of the lateral line. This superficial structure is a fish and amphibian mechanosensory organ, which is thought to be evolutionarily linked to the inner ear of vertebrates. Most of the mutant lines have been established in transgenic background expressing GFP or RFP in different cell types, which are composing the stereotypically distributed mechanosensory patches called neuromass. This right here is an illustration of a neuromass composed of supporting cells. These are hair cells, and the mantle cells are not illustrated, but bear with me. Use your imagination, and this would be the mantle cells. They compose the lateral line, which is equally distributed to the body of the zebrafish on both sides. We inject embryos with CRISPR-Cas9 and wait for them to sexually mature to inbreed. We follow their development by using fluorescent microscopy. We confirm the mutation by genotyping and sequencing. Once we confirm the mutation, we outcross them with wild types to obtain an F2 and later on an F3. This will give us a stable mutant line, which we will perform multiple experiments on to explore the role of the gene in hair cell regeneration. I really enjoyed my experience here in the lab, and I want to be clear, this is a young lab and it's full of young students, so they have their own dynamic and their own way of working. I very nicely integrated into it. I like my project. I would love to keep doing research here. I would encourage any student to choose development. That is why I chose development.